Buenas tardes a todos. Good afternoon to everybody. Thank you for coming to the last colloquia of the year 2021. So for me, it's a great pleasure to introduce you Michael Sadowsky. Michael Sadowsky ha estudiado bacteriología y microbiología en la Universidad de Wisconsin-Madison en Estados Unidos. A continuación hizo su doctorado en la Universidad de Hawái estudiando ecología microbiana y posteriormente ha incursionado en el campo de la microbiología, en la ecología microbiana molecular. Él ha estudiado comunidades microbianas en suelo, agua y en el intestino humano. Después de su doctorado en Hawái fue a hacer un postdoc a la Universidad de McGill en Montreal, Canadá. Después estuvo trabajando en el Departamento de Agricultura de Estados Unidos durante 4 años, antes de incorporarse a la Universidad de Minnesota el año 1989. Ha sido durante 12 años director del Instituto de Biotecnología de la Universidad de Minnesota y en estos días ha pasado a ser profesor emérito que mantiene la afiliación con la universidad y sigue muy activo en investigación. Es interesante destacar que también él ha trabajado fuertemente con los médicos y con organizaciones sin fines de lucro para tratar diferentes enfermedades humanas. Entre aquellas se destaca enfermedades intestinales e incluso el autismo, lo cual me parece tremendamente interesante. Entonces, thank you very much, Michael Sadowsky, for your accepting our invitation, and for us it's a great pleasure to can listen to you in this Microbial Colloquia Valparaíso 2021.
Thank you very much, Professor Seeger. I appreciate it very much, and I'm honored to be able to talk to you today about this new technology we've been working on. Well, it's not too new now. I've just been working on it 12 years, and how to treat human diseases with microorganisms, not drugs. I have to, there. I wasn't sure about the title, but understanding the gut microbiome helps us treat human diseases with microbes. So that's pretty much the focus of my talk today. And I'll be mostly focusing on the colon, which I call the final frontier for gut microbes. And I'll show you why I call it that. And then also there's light at the end of the tunnel that this type of technology can brighten our life by treating a variety of disease instead of just intestinal diseases. I wanted to mention that I would love to be with you in Valparaíso, but we have a Valparaíso in Estados Unidos, but yours is much prettier than ours. This is in the middle of the country, and it was named by a captain during the War of the 1812. So what I'd like to focus our discussion on today is called dysbiosis, and it's also referred to as dysbacteriosis, which typically means an imbalance of microbes in or on the body. Typically, the word is used to report conditions in the digestive tract that can be caused by microbes, and as you'll see in just a moment, this dysbiosis can be caused by a single microbial species. So in my talk today, rather than mentioning details of the scientific names, I'll be pointing to application of materials into the northern route, meaning the oral cavity, versus the southern route, meaning up from the bottom, as you might work. So we know quite a large amount of the intestine. We've been studying for nearly 100 years in much detail. We know that this important organ supplies the body with energy. It prevents colonization of pathogens, and it also is really important in the teaching of the body of the immune system. And that is by the induction and maintenance of the gut-associated lymphoid tissue. In this case, I'll focus our attention on one microorganism. It's called Clostridium or Clostridioides difficile, and its name means everything. It's very difficult. It's really an example of the distal gut organ failure due to microbiological devastation. And in this slide, you see the spore-forming bacterium Clostridium difficile with the refractile spore at its head end. Now, we know the colon is one of the largest organs in the human body. It's divided up into an ascending, transverse, descending, and then a sigmoid colon where it exits the body. It's the mucosal membrane surrounding the inside of the colon that is really important for its interaction with microorganisms and for its ability to move water in and out of the intestine to aid in digestion. Now, C. difficile infection I call a repeated saga of drugs and bugs. It starts very innocently with the consumption of antibiotics, who in some individuals will start destroying the microorganisms in the gut. Now, in your environment, our C. difficile spores are all around us, and given this denuded environment, the spores start to germinate, and when they do so, it leads to a production of a toxin, which causes severe diarrhea. Probably the worst diarrhea you can imagine. What do they treat this with normally is more antibiotics. So we have this cycle of antibiotic dysbiosis, antibiotics dysbiosis. And that's where this disease has a lot of problems. In the US, and I can't say for sure in Chile because I don't have that data, it affects about 2.5 million people a year. And initially it was thought mostly to be a hospital-acquired infection because every 13 out of 1,000 patients would get this. But this is increasing more recently in nursing and care facilities for the elderly. And in the US, the mortality is around 300 per day or 100,000 per year. So it could be a quite deadly disease. The number one risk factor for catching this bacterial disease is, of course, antibiotics, as I said. But people that have inflammatory bowel disease, are immunodeficient, or have HIV, or take gastric acid blockades, such as things that prevent acid from occurring in your stomach for reflux disease, could lead to infection. As to any way that you can enter, or the spores can enter the body via surgery or tube feeding. But by far, antibiotics is the number one. Now, this disease, I call it an extremely pernicious disease because there's a rule of thumb of the doubling risk. So if a patient gets this disease, they have a 20 to 30% chance of getting it again. And every time they get it again, the rate doubles on top of that. And eventually they have a disease called multiply recurrent C. difficile infection. It just keeps staying with you. And these people are very, very sick. So what do they do about this? Well, in the past, they've used tapered pulse therapy or narrow spectrum antibiotics. Again, treating the disease with more antibiotics. They've made some immunotherapy and there's some trials going on. They developed novel antibiotics, just target C. difficile. They've tried toxin binding resins and even probiotics. But generally, these have not worked well, especially for this multiply recurrent disease. So what do we do about it now? Well, we performed something that used to be called bacterial therapy, which we renamed intestinal microbiota transplantation. I wish I had this idea myself, but I can say it's an old idea revisited because about 1,700 years ago, the ancient Chinese reported that a physician was giving his patients yellow soup, which was actually made from dry feces containing microorganisms. A more modern rendition was published in 1958, where they talked about using fecal enema as an adjunct for the treatment of the disease at that time, which they called pseudomembraneous enterocolitis. More modern therapies involve nasal gastric tube administration or fecal enema. We focused our research on colonoscopy and capsule technology, and I'll explain a little bit how we developed some of that technology. So things have moved a little bit more forward, but the concept is quite old. The technique in its own right is fairly simple. You take a fecal material from a donor, and the donor could be any qualified individual, a relative or friend. And in our case, they have to pass a variety, about 64 tests. The material is placed in a blender. It's mixed with a buffer. It's separated using sieves or differential centrifugation. And then it's applied to the patient via this object in the lower right. And for the young people, you don't know what this is a colonoscope. And one of the channels in the colonoscope, besides being able to look at the colon, is an injection port where the material can be placed into the patient. I want to tell you about our first case, the index case, which gave us great hope in this technology. It was a 60-year-old woman who had chronic diarrhea for 8 months, continual. And during these 8 months, she was hospitalized because she kept getting dehydrated. And a colonoscopy showed ischemic colitis on biopsy, and you can see the image on the right, and that's done with a flexible sigmoid scope, and it shows a lot of bumps and polyps. She was going to the bathroom every 15 minutes and lost 27 kilos of weight and was confined to a wheelchair. Following what we called intestinal microbiota transplantation, or in the past fecal transplantation with her daughter's stool, she had no further problems for 2.5 years. She was cured within 3 days. The other problems came about because she subsequently took antibiotics, which destroyed her microbiota, which required us to give her some more, and I'll show you how that affects in just a minute. So now what we have, instead of bringing your daughter or donor or friend in, we have standardized frozen and freeze-dried microbiota preparations that we refer to as full spectrum microbiota. We don't separately culture any microbe. These are the purified microbes that come out of a stool sample. They're made via selective sieving, filtration, and differential centrifugation, and they produce about 99+% pure microbial preparations with a small amount of fibrous material. The technology I use is no different than that we've been using for the last 50 years to purify microbe from other compact matrices, such as bagasse or soil or aquatic sediments. And I'd like to tell you that all the stuff I'll be presenting to you today has been published and is openly available. So you can look up all these references as you wish. And feel free to write me if you have any questions. So who can be the donor? Well, not dogs, cats, or elephants, but really any person. But they have certain rules. They can't take any antibiotics in the last 3 months. They can't have any GI ailments. They can have no acute or chronic viral infections. They have to be an adult, and they have to go through many, many other diagnostic criteria, including weekly COVID-19 testing. So we rigorously screen these donors, and we have been approved by the United States Food and Drug Administration to produce these materials for use in patients to treat C. difficile disease. So I gave you a little bit of history. I'd like to tell you where we are now. We now have a University of Minnesota intestinal microbiota transplantation program. As I said, we have rigorously screened donors. We currently have 4. We produce standardized preparations of microbe material, and those are used for frozen or oral materials. I've also recently developed a pediatric material, and I'll tell you about that in a second, because they don't like to do anal injection on children, and most children can't take capsules. So we had to develop a new technology. So when we first started this research back in 2012, we realized that having fresh material is very difficult because you'd have to have the donor and the recipient at the same time. So we start to explore freezing material. And they're very, very close percentages of achieving our goal. And in this case, the success rate actually refers to curing the patient. So this is a phenomenal number, and it surpasses any cure rate by any drug known. Now the average for the material that's in a liquid form is about 96% cure rate, and that in a capsule form is about 89%. Still very, very high. So as scientists, of course, our main question is what's going on here? So we decided to use high throughput 16S amplicon sequencing and several other omics-based tools to provide answers to what's happening in the intestinal tract of our donors and recipients. So this is going to do this very briefly. But just to remind everyone, there's 3 domains of life that we know it now: the eukaryotes, including human, the bacteria, and the archaea. And I'm going to just focus the attention today on the bacteria, but we've also investigated the archaea and the fungi. But I'll just present bacteria results today. So what you have here is a histogram showing the microbiota presence in the donor on the left side and the patients before and after receiving the donor microbiota on the right side. And this was from a 67-year-old man who had multiply recurrent CDI, but he also had underlying ulcerative colitis, which is another inflammatory disease. As you can see, the donor is dominated mostly by 3 major groups of microbes: the Bacteroides, the Firmicutes, and a small amount of unclassified bacteria and proteobacteria, things like E. coli. The patient on the next bar, you could see, is totally dysbiotic, and its gut's been taken over by proteobacteria, probably leading to the symptoms that are seen. And the bar to the right of that was the patient after 3 days post-transplantation. And you can see how the patient now remarkably looks like the donor. Because of that, we refer to this phenomenon as engraftment. That is, the bacteria that we put into the patient are now engrafted into the host. I should point out that all these patients have been receiving antibiotic treatment prior to the engraftment with the donor's microbiota. So in a way, their intestinal tract is cleaned out and it's a clean slate for the new microorganisms to engraft, although we stopped treatment with antibiotic for 3 days prior to treatment. Here's another example. This is of a 74-year-old woman who had multiply recurrent CDI, and you can see she follows pretty much the same trend, although at the black line, the patient reverts back to what they were before due to taking antibiotics for urinary tract infection. And this is quite common in older women, that they get urinary tract infections. So we really tell our patients, please try to stay away from antibiotics as much as possible, unless absolutely necessary. And now we have capsule technology to treat them again. All these patients were treated in the hospital via colonoscopy. So to try to understand about the dynamics of the system, I hooked up my research group with that of Rob Knight, then at the University of Colorado. And I asked you to please go and look at this YouTube video that we made showing how the colonization of the gut occurs over time. And the patients very nicely agreed to give us fecal samples every day for the first week, every month for the first year, and then for 2 years following that. And we collected all that data and collated it. And you see a nice video of the re-engraftment of these microorganisms. So of course, as scientists, we're also curious, and how does IMT work? Well, the simple hypotheses that we started with were that commensal intestinal bacteria act as a barrier to pathogen establishment. But it could be more complicated than that. In fact, the commensal intestinal bacteria might also produce inhibitors that prevent pathogen growth. So we tested these hypotheses with the help of using metabolomics technology. I have to go back to a little bit of schooling for you to remind you about what goes on in the intestinal tract. And one of the most important things is the production and dissemination of bile. So bile, the primary bile acid, taurocolic acid, has been known for years to cause germination of spores, and is typically put in media, like for Bacillus and Clostridia, to allow spore germination. The body also produces something called secondary bile acids. An example would be deoxycholic acid. And this is known to inhibit germination of spores. And it's mostly produced, well, actually it's totally produced by microorganisms in the intestinal tract acting on primary bile acids. Now, the bile ducts carry bile from the liver, where it's synthesized, through to the pancreas, and it's released in the small intestine to aid in digestion and fat absorption. The remaining bile is stored in the gallbladder, and the body's pretty cheap in it, in that it uses about each bile acid about 20 times by recycling it back into the body. So the concentration of secondary bile acids in the intestine is fairly low because it doesn't stay there very long, it's recycled. So to test the hypothesis that changes in gut bile acid concentration is a mechanism behind IMT, we took patients that were treated and untreated, checked their microbiota, and then also looked at the presence of bile acids. And this is just a summary of published results. At the time, we collected the fecal samples and also sequenced the bacterial 16S ribosomal RNA. So, as I mentioned, the bile duct dumps primary bile acids, and as in a donor, which is a normal patient, we expect the primary bile acids to be low or relatively low in normal humans, because in the intestine, the microorganism that's converted. So you can see the primary bile acids in pre-FMT patients, and this is just an example of 3 of them, are very high. And in post-FMT patients, the primary bile acids are low. And just the opposite is true. Secondary bile acids increase during IMT. So the pre-FMT patients have hardly any secondary bile acids. And following treatment, they have secondary bile acids similar to the donor. So that most likely is one of the mechanisms, but not the only mechanisms for C. difficile, getting rid of C. difficile in the intestine using IMT. I mentioned to you previously that most of the early patients received IMT via colonoscopy, and that was done in the hospital suite. And I should mention, all we do is free of charge, except for the charge for the hospital suite. But it was difficult for the patients to get into the hospital, so we decided to develop capsule technology. And there's a lot of challenges with preservation technology, especially with freeze-drying complex mixtures of microorganisms. Although freeze-drying technology for microbes has been practiced for more than 50 years, but it's usually been done with the individual species. There's also delivery issues in getting the capsule, which typically are made to open in the stomach, to have delayed release into the intestine. And then, of course, there's difficulties with stability. Now, I want to go back to an idea that actually I didn't invent. I call it the babushka doll technology. And that's typically what you see with these toys from Russia, where you have one doll inside another doll inside another doll. So we decided to try this technology where we put a 0 type capsule in a double 0 type capsule to double encapsulate the forms to help them with passage through the gut. And that's the primary reason for doing this. We also use capsules that are delayed release, and they're made with a material that won't open up until the pH assumes normality. So they want to open up in the intestine. In our first test of 45 patients, 40 achieved complete clearance of C. difficile, about 89% success, with or without, and the PPI is the gastric blockade blocker. About 28 of 30 achieved clinical resolution from the first course of capsules, and those were at 2 times 10 to the 11th bacteria. And we checked variation in the capsule protocol, including washing the bowel out, changing the bacterial concentration, or the number of capsules. And all those did not significantly influence the outcome. Initially, we tried 4 to 27 capsules from 1 to 3 days. And now we just use 1 capsule, amazingly, and give it to them once, and they get clearance in 89% of the time. And again, this has been published as well in 2017, and you're welcome to look at this. Now, one of the questions that we had early on was this issue of stability. That is, how stable is the gut microbiota, as it passes through the stomach in a capsule for ultimate dissolution in the large intestine. But one way to do that is to use essentially a program called Source Tracker, which helps us follow individual microbes and follows over time points of days, weeks, months, and over long term. And using this technology, we're able to see that there's a slight difference between patients who receive fecal transplantation via colonoscopy, where there's days is where things resolve, to those that receive capsule technology, where things don't stabilize for weeks. So what is causing this engraftment delay? Is it inadequate freeze drying? Is it loss of viability? Is it loss of cell numbers? Is it bad technology for encapsulation? Or is it just because patients have different gut transit times? That is the time of material to make it from the mouth out through the anus. So to address some of these ideas, we did an experiment, and we compared the engraftment of freeze-dried microbiota that we rehydrated and gave colonoscopically or gave orally in capsules. And we used identical lots and doses. And this slide is just a very quick summary. And it's essentially telling you that capsule fecal microbiota technology is slightly delayed from those that receive by colonoscopy. And this technology also shows us that there's key groups of microorganisms that are slightly delayed, and that those microorganisms are essential for success. So it's most likely that the capsule technology itself is the problem. We know it's not the stability of the microbes following freeze drying. We know that it's not the numbers of microbes, because identical numbers are given via orally or colonoscopically. So it must have to do with the capsule technology, and we're investigating this right now. The take-home messages from all these studies show that the rapid engraftment associated with the cure is associated with increased relative abundances of the donors Lactospiraceae, Bacteroideaceae, and Polymorphidaceae. These are major families of microorganisms. And clinical failures are almost always associated with the relative loss of Bacteroideaceae, Lactospiraceae, and Ruminococcaceae, and then major microbiota families. So now we have an idea of what microbes are really important, and we can target target donors that have a large number of these microorganisms, such as Lactobacteroides and Porphyromonaceae, and try to see if we increase those in our donor pool, will we increase the frequency of cure. So I'd like just to summarize and leave a little time for discussion. I'd like to mention that recalcitrant C. difficile infection is the clearest example of the failure of the distal gut organ. Again, it's the largest organ in the body. The dysfunction of microbiota is likely common in many medical conditions, and this research is just the tip of the iceberg. The other take-home message that I think is important is that success of medicine in the 21st century is likely to come from restoration of healthy microbes. Just as in the 20th century, success succeeded by use of antibiotics. And this may lead to new therapies that do not involve any antibiotics. And that's why I say it's curing diseases with bugs and not drugs. As I was talking to Professor Seeger before, this technology is not only for humans, but has been practiced for quite a long time in animals. And we were actually called to 2 different zoos in Minnesota to treat monkeys who had severe intestinal diseases. So the first study we did was with a Brazil monkey called Jaziri. The recipient was not a human, but it was a donor monkey that we got from another zoo. We transplanted the material into this monkey. Unfortunately, the monkey actually had celiac disease, not Clostridium difficile disease. So we learned a little bit about that. But our most successful case is with Jeffrey's black-handed spider monkeys, where we're called to the local Minnesota Zoo, where 5 monkeys came down with C. difficile. 1 died prior to treatment, and we saved the other 4 monkeys using treatment with an adequate donor. So this technology works. And it's been used for a long time with cattle and other animals, and including dogs. But dogs, like mice and rats, are coprophagic and eat feces from their own animal types. So they actually do fecal transplantation by themselves quite often. So let me talk a little bit about the future directions of IMT. These include formal clinical trials. As I mentioned to you, we have investigated a new drug license from the FDA in the US. We're working with other universities and clinics in the US and nonprofit organizations in the US. Taxonomy is just a start in science. I showed you a little work that we learned about metabolomics with bile acid. We're doing metatranscriptomics, and we're focusing on particular groups of microbes that I showed you that have been restored, as well as people with failed IMT. We have new capsules I told you about, and we have pediatric preps that now, it's an oral preparation for little children to take. It's essentially ultra purified microbiota that are flavored with flavoring agents and added to a soluble material. And in addition to IMT being used for C. difficile disease, we also have trials going on with inflammatory bowel disease, metabolic syndrome, also known as obesity, irritable bowel syndrome. And we just finished some studies a few years back treating children that have autism spectrum disorder. Now, it doesn't cure autism. What it does is relieve intestinal distress that many autistic children have, which increases their good-naturedness, and they're not quite as active. And we have that in clinical trial now going on with Arizona State University. We're currently the sole suppliers of FMT or IMT materials in the United States, and we carry out all our own research and help patients for no charge at all. This is all part of our research program. So someday, I might say, you might be using foods that have these purified intestinal microbes contained in them. Just like now, where you eat things like yogurt, which have so-called probiotics in them to help aid in digestion, although the data on that is quite sketchy. So this is just a start of this type of technology. So I'd like to finish off acknowledging a lot of people that helped. I couldn't have begun this study and studies without the help of Alex Karutz. He's a physician, a PhD, MD, who I've worked with since the beginning. He actually approached me to ask if I'd help him in this project, and I immediately saw the benefits of doing so. And this, as I've said, is about 12 years ago. Byron Vaughn is an MD who's helping us with the ulcerative colitis studies. Raj Surajanan is helping us with the new capsule technology. He's in pharmacology. A lot of the work's done Chris Staley, who's my postdoc and now a professor here in medicine. And there's many, many graduate students, MD, PhD students, postdocs, vets, and the list goes on and on and on who contributed to this work. We couldn't have done this work without help from the hospital, and especially in the early days where all the nurses helped us. And then we have a separate facility at the university called the Molecular Cellular Therapeutics Facility, which produces all this material under guidelines conforming to the FDA requirements. And this is led by Murati Poe in our group. And now we're expanding this group to include 3 other people and opening up 2 separate rooms to produce the material in this high-tech facility. And we received funding over the years from the NIH, the university, Medical Foundation, Minnesota Medical Foundation, companies, and now more recently a nonprofit who are achieving cures together. So I want to thank you so much for the invitation, an opportunity to speak with you today. I wish you all much success and being safe during these weird times. I'd love to be visiting with you in Valparaíso right now, rather than the Valparaiso's here, because it's about -1 degree here today. So thank you again. It's been a pleasure speaking with you, and I'm happy to answer any questions you have.
Thank you, Mike, for this inspiring colloquia. Very, very interesting and challenging for us. So it's open for questions. Pueden hacer las preguntas en castellano y se las podemos traducir si es que lo requieren. No hay problema. So now Milko is in the room.
Hello, Mike.
So it's good.
Hola, Milko.
Long time without see you. I have you look very nice. You look nice. But in relation to your speech, I had a question. Is I know your research, but I'm very interested in the communication with bacteria. That in this study about transplantation, there's some study or some data about how bacteria not only like a barrier. You chose microbiome is a barrier in the intestinal barriers. You know, in communication in the intestine. And also, there's some study about the microRNA, bacteria microRNA, the action in this research. There's some study about that.
Yeah, so that's a very good question. So microbe communication is probably just as important as microbe-human communication. And the bile acids we reported are just the first chemical that we looked at that changed drastically in pre and post FMT patients. There's a variety of other signal molecules that we've not even looked at yet. We know what some of them are, but I think it's really, really important. Right? And that's an open area for other people to study. So we're talking about things like quorum sensing molecules, concentrations of very simple things like acetate, propionate. They all signal the host. In fact, that's the basis, we think, for issues with obesity, where the brain doesn't recognize that there's sufficient amounts of acetate and propionate in the gut. So I think you're exactly right. This is an open area. It's ripe for study, looking at communication molecules of the bacteria. We've tried to do it with the host a little bit, and we've done some initial studies on looking at patients pre and post FMT and the types and concentrations of fatty acids in the gut. It's not been very rewarding research, mostly because our technology is not great. But your second question was about microRNAs. We haven't looked at any of that at all. So again, that's totally open.
Okay, thank you.
De nada.
Yes, Mike, I'm very surprised that not surprised, but it's nice to listen that you're working with non-profit organizations in this field, because normally a lot of people are thinking about making big business, and you are working with the hospital and with this non-profit organization to treat people, normal people. Yes. So perhaps you can tell us which have been the nice aspects of this social investment in science, no, with not profit, but with the well-being of the people.
Very good. Thank you so much, Michael, for that question. So from the very beginning, my laboratory has practiced very basic as well as very applied research. And this was a great example of applied research with a direct outlet for need. And we saw that the need of this technology was massive. As I told you, in the US, it's millions of people each year getting this disease. Now, working with a physician and being a director, we both kind of looked at each other and said, God, we're so busy now. Let's not start a company. Let's just give this stuff away to treat the patients for free. So it's a different model than many professors take. Although I should say we patent this technology and license it to other companies. We didn't want to be involved with running our own company, and we didn't want to be a for-profit organization. So that's why we hooked up with these nonprofit organizations that provide us funds just to produce the microorganisms and distribute them. We're not taking any salaries. It's just for the production purposes only. So it's a different model, and one that Alex Carus, the physician, and I think is really important to get the science out to the people as soon as possible.
Exactly. This is something that is now challenging in Chile. So the people are expecting that the science will contribute much more to the social welfare and that to to the society that will benefit a lot. And in Chile, things are changing, so we hope that science will have more importance in Chile and that we can apply really science to improve the quality of life of the people. And this work with non-profit organizations is a different paradigm, because here the last years we have been pushed to do new companies, and I understand that companies could be very interesting, but I think also that when the people see that this could be without profit and that could be massive applied, that sounds very, very interesting. I want to make you another question. If they, perhaps there are some questions in the public.
It couldn't have been that clear.
Okay. No. Yes. You speak about these 3 types of specific bugs that are very important for the health of the intestine, and perhaps you can tell us a little more about these bacteria, which functions of these bacteria could be involved in this recovery from the disease. And I don't know if you have looked about the metabolic genes or the genes that could be also involved with this. They produce perhaps some compounds that inhibit bacteria that are not so good for people, or what are they doing, these 3 bacteria types that you remarks?
Yeah, so I can say it very succinctly, we don't know. The hypothesis is that these organisms need to fulfill and fill a niche in the host that prevents colonization by C. difficile. That's the simple answer, although we have no data to support that. We do know if these organisms are present that the patient doesn't get the disease or is cured the disease. I want to mention, though, that the story is a little more complicated than I presented, because C. difficile resides all around us all the time. And some people get the disease and some people don't, and we don't know the basis of that. We also know that when we go into the households of our patients, we can detect C. difficile in 100% of the households of patients with C. difficile. It's in things like carpeting, in the toilet, on the handles in the bathroom. It's all around them. So some of our patients might also be getting re-inoculated, but it's really important that these 3 groups are there. And now it's for you people to figure out, the young people to figure out what those organisms are doing. And part of the problem is the taxonomy is very broad. So when I say Lachnospiraceae, that's a lot of organisms, right? So we have to use different technologies now, which is full metagenome sequencing, to identify the individual microbes. We've also used some array technology, I didn't present that data, using individual gut microbes on a chip, and we now have an idea of which microbes are important, but we don't know what they're doing.
Okay, thank you, Mike. More questions? When I listed you, Mike, and I listed what are doing the engineers with the water treatment, the people that work with water treatment always speak about microbial communities, and they are not interested in to isolate bacteria or archaea because they think that the whole community is doing the work, you know? So in this case, you also say that you isolate you don't isolate the bacteria, you take all the microbiota from the feces and you inoculate the complete microbiota to the people. I think this is a very different approach, and it's very interesting that it's not looking that you have only one bacteria that you can inoculate and that will cure the people. Or you take the whole community of a donor that is healthy and you put it inside. So it's a much more holistic view of treatment, and this is very nice. I don't know if you can comment some about this.
Yeah, absolutely, I can comment on it. So I have to remind people that an adult human intestinal tract contains 700 to 1,000 species of microorganisms, present anywhere from approximately 10 to the 4th to 10 to the 12th bacteria per gram of feces. So it's a huge range of numbers and a huge range of types. And when people ask me, why didn't I take the culture-based approach? I say, because I'm pretty stupid, right? I'm not smart enough to do that. I don't know which organisms, what ratio, if I can even grow them. And people that have tried to inoculate humans with even a single microorganism have failed to do so due to inoculation resistance. So it's for that lack of knowledge and our inability to figure out what the ratios are, which organisms need to be there, which one first, can I grow them? Can I grow them in large numbers? Can I grow them pure? We decided to actually take this approach, right? Where it's more of a holistic ecological approach to solving the problem. There are companies out there that have tried growing individual microorganisms and pulling them together. Some have been somewhat successful in treating C. difficile disease when the organisms produce some antitoxin. However, those patients typically fail over the long term, and all the clinical trials where that's been going on have been ended.
Okay, great. So we have a question of Francisca Martinich. She asks you, do you always need a real donor, or can lab mix be used as a donor?
It doesn't matter who the donor is, whether it's a lab mate or not, they all have to go through the serious testing that we do. So our donors are all volunteers now. They have to come to the university and make, I call it, deposit in a special bathroom at the university that we keep very clean. And again, before they can become a donor, they have to go through this rigorous screening. It could be a student, it could be your friend, your relative, it doesn't matter. They all are treated the same.
Okay, thank you. So is there more questions? You can make the questions by the chat if you want, or you can do it on Spanish, whatever. Francisca, are you happy with the answer?
Yes, thank you.
You're welcome. No other questions? I can't believe it.
Beatriz or Roberto.
Paulina tiene una.
Paulina Vega, she has some questions. Yes, I think so. So I have to say, Mike, that normally we have the colloquia on Spanish, most of the colloquia on Spanish, and sometimes the people feel that the English is not good enough. But
yeah, mi español es muy malo.
Yes.
Mike, I had a question. I'm thinking about the experiment with the monkey,
Yes.
spider monkey. What is the idea to use this technology for the zoo protection, environmental protection, wild type or animal? What is the final focus? They try to use this technology in animal.
Well, for many animals get dysbiosis a lot. You know, they get vomiting disease, they get diarrheal diseases. So for animals such as cows, this has been mostly used to treat obvious disease. Right. It's also been used, as I said, in dogs as well. For us, we specifically aim for a specific disease, but we have received some phone calls and some emails from some veterinary manufacturers of medicines asking us to make encapsulated material for people to treat their animals. And we're investigating that right now. The problem is we need a separate facility because we can't produce an animal-based product in the same facility we make a human-based product, but the technology is the same. So yes. in that case, it's for prevention.
Yes, but this technology is thinking to pet or for conservation animal or zoo animal?
It's for pets and zoo animals right now.
Okay.
Yeah, but also, you know, for cattle, which are used for production, you know, it has been used in there as well, but we're not marketing it for that.
You know, now the world is changing. Some animal is under extinction, dangerous. That maybe there's some problem with increase the new disease. That maybe another area can be this in the conservation of the wildlife.
Yeah, and if people ask us to make material, we can easily do so. The technology is the same no matter what feces you use. So
have you tried from human to animal?
I'm sorry, what's the question?
from human to
no,
animal to human.
we don't do any.
You can mix, huh? You can mix.
We don't do trans kingdom or trans family transplantations. Yeah. I think that might be a little unethical. Yeah.
Mike, I have another question. We was speaking also before the colloquia about what you told, this treatment of autism. So autism is a very, very important in Chile and everywhere in all the world. And we have increasing numbers of autism in Chile and in other countries also. And I find very interesting that these diseased people can be treated when they are children because they have problems of the intestine. And perhaps you can comment more about this, because I think this is a absolute open field in Chile. Nobody knows, I think, about what you was talking, that you can also use microbes for this. And okay.
Yeah, absolutely. So we were contacted by a professor at Arizona State University, Professor Jim Adams, who's been studying autism for years. And when he read about our work curing C. difficile, he asked us if we could help him seeing if this would work for at least making the children that have autism spectrum disorder feel a little better, so they might not act out. So we did a very small open label study. That is that the people giving the material knew what was the control, what was the experimental, and we got approximately a 40% success rate in improving the behavior of autistic children using this technology. And as I mentioned to you, at the time, we didn't know how to give children the material, so we actually dissolved it in chocolate milk and gave it to them. But even with a 40% success rate or 39% success rate, and these are improvement in behavior as reported by the parents. So we weren't involved in determining this, which reduced some of the bias. We had success. So again, the idea is that you're not curing autism, you're changing the behavior of children that might have discomfort due to intestinal ailments, which many autistic children have. So currently now we have a closed label, placebo-controlled trial going on through Arizona State University and several other centers around the United States where we're supplying material to them. So I guess the jury is still out on this closed label study, but the initial results were very encouraging. So again, it shows you that this technology can be used not only for C. diff, but for autism. I think the technology has great use in treating oral diseases and skin diseases as well. So this is a great area that's ripe for experimentation. Each one has a separate ecological system to study. So, it's like the low-hanging fruit in all this work is there for you to take.
Fantastic. No, this for me, it's very fantastic. So we will make an agreement between Chile and University of Wisconsin. Perhaps we can have the opportunity that some people from Chile can also come there. And I find this really amazing. I want to do another question, because Clostridium difficile is a big problem everywhere, of course, but also in Chile. And why Clostridium difficile is so successful in the hospitals? So a lot of people get this illness in the hospitals, I think. So why they are able to survive so well in the hospitals and propagate in the hospitals?
I'm sorry, are you talking about obesity or you're talking about?
No, Clostridium difficile. Clostridium difficile is very...
Okay, sorry.
Yes.
Yeah, so I mean, hospitals have been targeting this for a while now. I think they are getting much better at sterilizing the environment around them. Clostridium spores, as is bacillus spores, are extremely durable and able to survive in really extreme environments, including the deserts of Chile as well. But the hospitals are getting better, and I think a lot more of the patients are getting infected at home now or in nursing care facilities. And that's mostly due to the fact that the spore hides in carpeting and in fabrics. And we have a paper we just published last week showing how you can kill the spores in carpeting and fabrics. And that came out of our research looking at the households of patients with C. difficile. The #1 location to find the spores in their house was in the vacuum cleaner. So it's definitely in the carpeting and the fabric. So I think the hospital issue is going to be moving away from the hospital because they're being much more clean now, and they're moving to the home and to the nursing care facility. Again, cleanliness. If you clean everything with hypochlorite-based products on surfaces, it will get rid of all the spores. But you can't put that on carpeting or fabric, it'll change the color.
Okay, very, very interesting. So we will open for the last time the questions to the public, if there is any question.
Michael, hay tres preguntas en el chat.
Ah, sorry, gracias, thank you. No,
Karen,
here we have the questions, yes.
Karen, Paulina y
Yes,
Gonzalo.
yes. Okay, here yes, there are questions in the chat. I was very lazy. So the first question comes from Karen. She says the donor needs to eat some special diet before, and I would like to know if this kind of approach can be used as a treatment for lactose intolerant people.
Okay. So it doesn't matter the diet of the donor. They don't need anything special. We have success with people that are vegan, people that eat lots of meat, people that are pescatarian. It doesn't matter, right? It's just a healthy gut is a healthy gut, okay? And we don't separate bacteria groups saying, oh, you're vegan, I only give you vegan host. It doesn't matter, okay? Now, whether this can cure lactose intolerance is probably doubtful, because the intolerance is typically due to an enzymatic deficiency in the host themselves, in their ability to cleave lactose. So lactose is fermented by bacteria into acid and gas, and those individuals that are intolerant have problems with their digestion. So it would be solved, although we haven't tried that.
Okay. Yes, Karen say, I am a veterinary and we usually use this approach with rabbits and guinea pigs, which have dysbiosis, but we give them directly by the mouth.
Mm-hmm. Yes.
Okay.
So rabbits, dogs, rats, mice, they're all coprophagic, and they have no problem in eating feces from other animals. So I think that's pretty easy to do. I never thought about rabbits before, but yeah, it should work in all animals as a technology.
Okay, thank you very much, Karen, for sharing your knowledge with us and for the questions. So Paulina Vega is also asking. She say that she have seen that with rats, some of them they are very obese. They can be treated with microbial intestinal microbiota, and then they can get more slim.
Yes.
And if you have any capsules for this purpose.
Yeah. So we tried, we did some initial studies that received a small amount of funding to look at this issue of obesity in humans. And as you correctly mentioned, this has been studied mostly in mice and rats, although a lot of the research is not translatable. But I think in this case it is. The hypothesis is quite simply that a lean individual produces adequate amount of acetate, propionate, and other fatty acids to supply the host with energy, so they don't need to eat a lot. And that energy, those chemicals feed back to the brain, telling the brain that I'm full, I had enough. And individuals that are obese have issues with that communication. So the initial studies we've done look very favorable, although large open, closed label studies, to my knowledge, haven't been done. The concept's quite straightforward to do. I just don't know where it is right now. As I said, we did some initial studies that look promising, but the results are difficult sometimes to interpret, except for two things. That is weight gain and what's called glucose intolerance, their ability to metabolize glucose. And both those look favorable from our initial studies. So yes, this is going to be an area that you're going to see a lot more research in. I just caution you not to take those over-the-counter medicines you see that says, oh, take this, you'll lose weight, without really knowing what you're taking.
Okay, great. So here is asking Gonzalo Jorquera. He say many intestinal bacteria are anaerobic. How do you prevent their death when you obtain the sample from the donor because oxygen can kill them?
Yes. So you're exactly right. We typically do our first set of isolations under partially anaerobic conditions. We felt that the most dangerous part for the microorganisms would be blending in a blender where air might mix into the system, a lot of air. So in fact, all our blending is done under nitrogen gas. So I use commercial blenders and modify them to have inlet ports. We also now have the technology to do all our centrifugations under anaerobic conditions. Ultimately, once the organism is freeze-dried, it's protected, obviously, because it's inanimate in a way. So those are, we think, are the most critical steps, the centrifugation and the blending, and those are done under nitrogen gas.
Okay, that's very important. Thank you very much, Gonzalo, for the question. I don't know if there is another question by the chat. I don't see any other question by the chat. Gonzalo and Paulina, it's you have any other question, or Karen? Okay, so I think this is an open talk. So we hope that Mike, when you come to Chile to visit Milko in the nice south, you also can pass to Valparaíso. That is a very nice harbor, and Milko knows. And we can have here a very nice swim in the sea, and have a nice dinner or lunch in front of the ocean. And we can keep in touch. So we are very interested to keep in touch with you. So Milko have this strong links. Thanks to Milko, we have this contact with you, and has been very great to listen to you. And thank you very much.
It's been my pleasure to meet with you today, and I'd love to meet you in person in the future. And then I can eat some good Chilean mariscos.
Yes, we are thinking to